Hello again. Right, this is the third part, I might do a fourth, of meiosis and the explanation of meiosis. And if you remember back to the second video, we answered part of this question. How does meiosis lead to increased genetic variation? And we discussed crossing over in the first part. Second thing that happens is a process which is called, it's got a few different names actually, but on the AQA biology we tend to refer to it as independent segregation. But you may see it in other textbooks or online elsewhere as random segregation, random assortment, independent assortment. All of those things tend to mean the same. We are going to refer to it as independent segregation. Segregation, I'll spell that right there. Okay, so just a quick overview of where we're up to at this point. Here we can see a diploid cell. So diploid cell, 2N, and it contains two, I hope you can see this, pairs of homologs, two homologous chromosomes. You can see the longer ones that are a homologous pair and the shorter ones which are homologous pair. Just to reiterate again, a homologous pair, two chromosomes that contain the same genes at the same loci. Now, actually you would never see a cell looking like this because the chromosomes only become condensed and visible under a light microscope during prophase, which is after DNA replication. What you might, however, see is something like this. So between this stage and this stage, DNA replication has taken place. Now this is prophase 1. If you remember back to the last video, we remember that prophase 1 is the first stage of meiosis, and that is when crossing over may take place. Okay, there are some questions that will come up, particularly related to inheritance in the second year, specifically when we look at autosomal linked genes, where crossing over is part of the answer. Crossing over is actually quite infrequent or rare, and that's sometimes a marking point. We'll come back to that in a bit more detail when we when we start on inheritance later on in the year and into the second year. Okay, this next event then, independent, sorry. It's been a few weeks since I've done this. Independent segregation takes place in metaphase 1. So this is still part of that first meiotic division. Prophase 1 comes first, then metaphase 1. And metaphase, you will remember from mitosis, is the period of cell division when the chromosomes line up across the equator, or across the middle of the cell. So think about metaphase for middle. When we see those chromosomes across the middle, we know it's metaphase. Little difference here between metaphase in meiosis and metaphase in mitosis, or metaphase 1 in meiosis and metaphase in mitosis. They line up across the equator, still in there, homologous pairs. Do you remember during prophase, they pair up, the homologous pairs pair up to form bivalence. Crossing over may take place. After crossing over perhaps has taken place, they migrate to the middle of the cell, across I think of it as the equator of the cell. Now, the way that they do this is completely random. Okay? This is where we get this idea of it being independent. So there's no organisation of whether or not a maternal chromosome is on the top and a paternal chromosome is on the bottom, or anything else. Okay, so it's completely random. If this is, let's just for the sake of it, let's say that this one is paternal above the equator for that larger chromosome, and paternal below the equator for the smaller chromosome. Oh dear, my blind is just broken. But anyway, we'll carry on. Like I say, that is completely random. It could be any orientation of those chromosomes across the equator. The product of that, well, because it's random, there are a number of different outcomes that could potentially happen. So I gave you a phrase that I use for crossing over. I said during crossing over, the maternal and paternal alleles are exchanged or mixed. For independent segregation, I use a similar sentence, but it's slightly different. I said during independent segregation, this time the maternal and paternal chromosomes are mixed. Okay, because during metaphase 1, the maternal and the paternal chromosomes may orientate themselves either above or below the middle of the cell, which is completely random. Just give me 1 second, I'll see if I can get that line back up. Quite a bit of climbing. Alright. Hopefully that'll stay there. Okay, so the maternal and paternal chromosomes are mixed. The outcome of that is you get a random assortment, a random mix of maternal and paternal chromosomes in these gametes. So all of these are gametes, and there are different possible combinations of those chromosomes. So you may end up with a maternal longer one and a maternal shorter one together. If that's the case, you could end up with paternal and paternal, or you might end up with a mix of each. So perhaps a paternal longer chromosome and a maternal shorter one. Alternatively, the fourth possible combination would be a maternal longer one, paternal shorter one in those haploid gametes at the end. Now there's a way of calculating the possible the possible combinations of maternal and paternal chromosomes, and that's using quite a straightforward formula. 2 to the n gives us the possible combinations. So where n is the number of homologous pairs. In this cell that we've started with, there are 2 homologous pairs. 2 squared, or 2 to the power of 2, is 4. 4 possible combinations. If there were 3 homologous chromosomes, and that's, I think in the diagram in the notes, there are 3 possible chromosomes, so 2 to the power of 3, or 2 cubed, would be 8 possible combinations, and that's what's shown in the diagram. In humans, so humans have 23 homologous pairs. So 2 to the power of 23 is 8 million. Oh no, I should have written this down. 8,388,608 possible combinations. Unlike Priti Patel, I was able to read that number out properly. Bit of politics. Okay, so 2 to the power of 23 gives us, well, nearly 8.5 million possible combinations of maternal and paternal chromosomes from each one of those homologous pairs. And that's an enormous number. And what you've got to remember, that is nearly 8.5 million possible combinations of maternal and paternal chromosomes, let's say in a sperm. There would also be 8.5 million possible combinations in an egg. Multiply those two things together, and you get an enormous number, 70 million possible combinations of those chromosomes from the mother and father. And that increases the genetic variation. If you then combine that with the possible exchange of alleles between the maternal and paternal chromosomes during crossing over, we've again increased the genetic variation. Many of the questions that you get on meiosis will relate more specifically to these ideas of how meiosis leads to genetic variation, rather than the events that are taking place during each of the stages of meiosis. It's important to know these two, I suppose. So during prophase 1 is where we get crossing over. Sometimes it's infrequent, but it can happen. And during metaphase 1 is where independent segregation takes place. Your focus should be on understanding how that increases genetic variation. So independent segregation mixes the maternal and paternal chromosomes. Crossing over mixes the maternal and paternal alleles. This is really important for showing all of those possible combinations and giving us this massive number for this genetic variation. Let me just check. I think that'll be everything for this. So, one last thing I will say. Meiosis increases genetic variation through independent segregation in metaphase 1, crossing over during prophase 1, producing gametes which have a huge combination of maternal and paternal chromosomes, perhaps new combinations, recombinants of maternal and paternal alleles, random fusing of those gametes during fertilization is another reason why the genetic variation increases. That's slightly separate to meiosis. Meiosis produces the gametes, fertilization obviously happens afterwards. Okay, thank you.